Alright, we're continuing our series on fourth quarter grade 12 biology. So this is going to be part 2. So let's talk about gram staining. Sorry. Okay. Gram staining. This is so for gram staining, it classifies bacterial species into two groups based on the differences, particularly in the cell wall composition. So this is an important detail. So the cell wall composition is different for the two groups. So gram positive, the cell wall is simpler. Gram negative, the structure is more complex. And yes, there's a reason why it's important to determine whether a particular infection is caused by gram positive or gram negative bacteria. For gram positive bacteria, um, it is easier to treat because it lacks a protective outer layer. Unlike gram-negative bacteria, because they have a complex structure and there is a low amount of peptidoglycan, these two things come together. So basically, it is harder to treat. It impedes the entry of drugs. That's the tendency. So yes, regarding how you determine whether a bacteria is gram positive or gram negative, it has something to do with the safranin and there's another solution. But basically, um, for gram positive bacteria, the color is purple or blue. It traps crystal violet in the cytoplasm, masking the added red safranin dye. And for gram-negative bacteria, it easily rinses crystal violet from the cytoplasm, and the cell is colored pink or red. So I'm not sure if I can still remember which between these two groups remain or don't change color after being exposed to the last solution in the gram staining process. I'm not sure. But these are the details that I have in my reviewer. So, yes, samples are stained with crystal violet dye and iodine. So this is the first process. And the samples are rinsed in alcohol. They are stained with red dye, for instance, safranin. And from there, you quickly determine if the infection is done by gram positive or gram negative. So, what are the effectiveness of certain antibiotics? Antibiotics, by the way, are intended to help in inhibiting peptidoglycan cross-linking. So it has something to do with uh, the peptidoglycan content in gram-positive and gram-negative. So by inhibiting peptidoglycan cross-linking, bacteria, regardless if they are gram-positive or gram-negative, are not able to uh, continue their or carry out their reproduction or reproductive processes. So, the resulting cell wall may not be functional, especially for gram-positive bacteria, because again, for gram-positive, the cell wall is simpler, and it's easier to treat overall. One famous antibiotic is penicillin. This was most especially used during World War II, just for historical background. So it derives from the inhibition of peptidoglycan cross-linking. Thirdly, it destroys many species of pathogenic bacteria without adversely affecting human cells. Why? That's because antibiotics only target bacteria or entities that contain peptidoglycan. So it only concerns bacteria which have peptidoglycan. Human cells are not affected because they don't have peptidoglycan. So that's basically it, the logic of it. And let's go here. Myobacterium, I think TB is tuberculosis. So It has virulent strains, and it is resistant to one or more antibiotics. So that's just for one example. Um, I think these are parts of a bacteria, I suppose. The capsule is the thick layer of polysaccharide or protein. Um, it covers the prokaryotic cell wall. Yes, I remember. Yes. So aside from a bacterium having a cell wall, a capsule encompasses it. So it's not the cell wall that's in the outermost layer. Cell wall also protects against dehydration, and it shields from attacks by the host's immune system. Sorry, I'm not sure what's going on. Okay. Um, Motility. This isn't exactly a part of a bacterium, but what basically motility means is the ability to move, um, similar to the adjective motile, um, the ability or capability to move. So they say that bacteria move at a speed of 360 kilometers per hour. I'm not sure if that's actually correct, but there's a reason why I wrote that anyways here. Um, Everything in this reviewer is based on research. So yeah, that's one thing that we can vouch on. 50% of prokaryotes can move. That's for sure. How about fimbriae? Fimbriae is in other words called the attachment pill. They are shorter and more numerous. Sorry, uh, there's no S. They allow prokaryotes to stick their substrate, uh, or basically stick themselves to one another. Um, it's associated with the sex pili. The sex pili pulls cells together before the DNA transfer. So I'm not sure if this is correct to say, but in a sense, the sex pili in bacteria um, is similar to the mycelium. Is it mycelium? I think it is, yes. The mycelium found in fungi. So before before connecting uh, one fungus to another compatible fungus, the mycelium is the one that's responsible for determining the compatibility. So yeah, just for extra reference, I said that. Flagella. Um, Scattered all over or concentrated on both or one end. So flagella is basically the tail. Um, if you still remember uh, when I mentioned about the sperm cell, so basically the tail is the flagella. That also goes. That's also applicable in the case of bacteria. So this is the bacteria. This is the capsule. This is the flagellum. And it can be more than one flagellum for some reason. Okay. Um, where do we start? Uh, Aerobic and photosynthetic prokaryote. So what's the difference? Well, these prokaryotes have specialized membranes that perform metabolic functions. Speaking of metabolic functions, particularly for bacteria in general, um, the it is recognized that bacteria lack um, organelles such as the nucleus and other organelles that are responsible for performing metabolic functions. The mitochondria is one of them. So those things, the Golgi apparatus, lysosomes, uh, bacteria lack them. So there's a question: how do bacteria perform metabolic functions in the absence of these kinds of important organelles? Usually, they're only found in animal cells, plant cells. Well, the answer is basically their plasma membrane. The plasma membrane of bacteria is very complex, and it compensates for, uh, I suppose, everything. Maybe it performs the metabolic functions in a different way, but nevertheless, the plasma membrane is the one that's compensating for it. That's the important detail. So, yeah. For aerobic prokaryotes, respiratory membranes, photosynthetic prokaryotes, thylakoid. From the idea of photosynthesis and thylakoids, those two words are usually associated with each other in the context of photosynthesis. Photosynthetic, thylakoid, that's related to plants. Aerobic, um, respiratory. Just like people, we have respiratory systems. Although I'm not sure if it's actually literally respiration for those prokaryotes that are aerobic. Okay. Taxis. This is not what you usually know in common context. Taxis, it refers to the movement towards or away a stimulus. So, chemotaxis basically change movement pattern in response to chemicals. So, if it's a positive chemotaxi, um, basically there is a movement towards nutrients or oxygen. And if there's a negative chemotaxi, uh, there is a movement away from the toxics. So, okay. Less DNA consists of circular chromosome. There is no membrane bound nucleus, like what I said earlier. Um, Instead, there's a nucleoid. This nucleoid houses the chromosomes, the genetic material, and the plasmid. This is the ring, a ring of DNA that carries a few genes, and smaller ribosomes are different from eukaryotes in protein and RNA content. They allow certain antibiotics to bind to ribosomes, block protein synthesis in prokaryotes. That's basically the end result. So, in the inability to carry out protein synthesis, prokaryotes may not continue their species, especially if these bacteria are harmful to the body. And that's the reason why antibiotics are useful. They address particularly uh, that kind of dilemma. Okay, reproduction and adaptation, highly successful and increase in potential reproduction, uh, fast in favorable environment. I'm not sure what that means, but um, maybe it's trying to say that prokaryotes are useful. They adapt well in favorable environments. Binary fission. Many prokaryotes divide every 3 hours. So there is basically a new generation every 20 minutes. What are the reproductive features of prokaryotes? So they're they're small. They reproduce by binary fission and they have short generation times. What contributes to the prokaryotes' success in surviving harsh conditions? We answered earlier how they're able to compensate for the absence of organelles, membrane-bound organelles in performing metabolic functions. Right now, let's answer how they survive harsh conditions. So there's something called endospores. These endospores are resistant cells exhibited by prokaryotes when nutrients are lacking. Original cell forms the endospore. Basically, the water is removed and the metabolism halts. So basically, prokaryotes enter a state of dormancy, and when conditions are favorable again, that's when they return and go back to carrying out metabolic functions. It's similar to how plants, such as cactus, um, they close. I'm not sure what they usually close. What's called in what they close, but In order to conserve water, they close that particular organ, part of that plant, so that no water is wasted and no carbon dioxide is received in exchange. But yeah, so the original cell disintegrates, the endospore is left, and um, there is a dormancy. Uh, dormancy occurs, but viability lasts for centuries. They can rehydrate, resume metabolism when the environment is more favorable once again, or if it improves. Okay, so an increased level of genetic diversity in prokaryotes. So we have rapid reproduction, mutation, and genetic recombination. Let's tackle each one of them. So for rapid reproduction and mutation, so prokaryotes are classified um, into two groups in terms of how they reproduce. So we have prokaryotes that reproduce by sexual reproduction, and the other group performs asexual reproduction. So for those that perform sexual reproduction, uh, there's a generation of novel allele by new mutation. So this is rare at any gene. There are three methods. For asexual reproduction, there is an increased amount um, of genetic variation, and this is a result of mutation. Let's go to recombination, genetic recombination. There are three methods. We have transduction, transformation, and conjugation. For transduction, the genotype and phenotype of a prokaryotic cell is altered by the uptake of foreign DNA. I wasn't able to cite an example for this. For transformation, this is a type of horizontal gene transfer. So for instance, bacteriophage, viruses infecting bacteria, they carry basically bacterial genes from one host to another. How about conjugation? Conjugation. Genetic material is transferred between two bacterial cells that are temporarily joined. So DNA transfer is one way. Donors use sex pili to attach to recipient. So basically conjugation is associated with the employment of what? Wait, this one, the sex pili, what I mentioned earlier. Okay, now diverse nutritional and metabolic adaptations. Prokaryotes can be organized, aside from reproduction, they can also be organized by nutrition. So, um, nutrition diverse prokaryotes are greater than those of eukaryotes, those of their eukaryotic counterparts. Okay, I suppose I skipped this part. Double cooperation, nitrogen metabolism, role of oxygen in metabolism. Okay, we're finished with prokaryotes. Let's move to protists. Actually, wait. I'll end this video. Let's dedicate the next part for protists. Thanks for watching.